Hi, everybody. We wanted to go ahead and get started. Thank you for being here. This is a great crowd for our first in a series about descriptive epidemiology research in DCEG. We're going to be holding this seminar series every couple of months, so we're hoping our next one will be perhaps in early January. The goal of this seminar series is to highlight descriptive epidemiology research in DCEG, including data resources, analytic tools, and the research itself, and hopefully encourage more descriptive epi research in the division. And we're defining descriptive epidemiology to include cancer registry research, of course, as well as linkage studies, medical claims data, national surveys. So we're taking a broad approach to descriptive epi in this seminar series. In addition, we have a working group, and if anybody is interested in participating in that, please email me, and I'd be happy to add you to our listserv. So our speakers today do not need an introduction, but they deserve one. So we're going to have Stephen come on up and introduce them. Thank you.
All right, there are a lot of cameras around here, and the first thing I would say is that those who want to tweet can tweet. The current speakers are not opposed to that. But really, on a serious note, it's my great pleasure to be able to introduce two great living legends of cancer epidemiology, Joe Fraumeni and Bob Hoover, who have graciously agreed to talk about the history of cancer epidemiology and the historical perspectives and really where the field has come from and where they're going. And I think, you know, we here at DCEG really look at the two of them and see them as great giants. And Isaac Newton said, if I can see further, it is by standing on the shoulders of giants. But Isaac Newton was not the person that first said that. John of Salisbury in 1159, a very famous theologian, made this comment in understanding and coming closer in a religious sense to God. You stand on the shoulders of giants to be able to better understand. And I think that today's seminar is really an appropriate opportunity for us to hear what the giants have done and what they think and what they see, because so many of us around this room have worked with them and are continuing to work with them, and particularly the next generation, standing on the accomplishments and the foundations that they have really put in place. And the key issue here is really what's past is prologue, as Shakespeare said in his final play, The Tempest. And I think what we know, what we've learned so far, what? I know, okay. I know, but it's famously said in Act 2 of The Tempest. Thank you, Mark, and we'll have a discussion of that a little bit after in the question and answer session. But again, it is really a great pleasure to welcome them to the podium, and I think that the so-called Joe and Bob team will come forward and present in the order in which they have arranged this, and it's really wonderful that Meredith and the group have been able to bring them to the podium to really share with the rest of us their perspectives. And it's without further ado that we welcome Bob and Joe. And of course, I also want to say we welcome Joe's wonderful wife, Tricia, who's here as a guest. And we see several other distinguished individuals who've worked in and around DCEG over the years, Bill and company, and it's really wonderful to have all of these people here, as well as our next generations in front of us. So, Bob and Joe, please, the stage is yours. Thank you.
Thank you, Stephen. Thank you, and I want to thank you all for coming too, and actually I'll single out Bill Blott here because we're going to show a lot of what he was mainly responsible for when he was here. So this should be, we're hoping that this is going to be fun for everybody. It'll be certainly fun for us. As you'll notice, we're in a different kind of position for presenting a lecture here. We're sitting down today, and you know Joe has some issues, but they really are actually another example of the font of wisdom of Joe Fraumeni. That's a font of wisdom that I have drunk from for 45 years. And in the most recent instance, he actually has taught me that when you get to a certain age, it's probably not a good idea to join your nephews on a basketball court to show off some of your Kobe Bryant moves to the hoop. We should, Okay, so we'll start with definition. This is one of the simplest definitions, but I think one of the originals, but it works, that descriptive epi is a description of the distribution of disease with comparisons of the frequency in different populations and different segments of the same population. And it is usually followed by analytic epidemiology, which are straightforward observational studies designed to specifically examine the hypotheses as a result of this. While descriptive epi has been around for centuries in the infectious disease world, John Snow making his maps and then deciding to take the handle off the pump, but for chronic disease epidemiology, it didn't come along until quite a bit later. And actually, the interest in descriptive epidemiology was promoted avidly by Sir Richard Dahl. You probably can't read all of the things on here, but he talked about the search for clues for hypotheses, with one of the most rewarding is the comparison of the frequency with which a disease occurs in different communities, in different areas, and in different times, basically descriptive epidemiology. He also provided us with a marvelous example of what it can do. This is his famous study in the British Medical Journal in 1950 of lung cancer, and between this one and the one by and another one done in this country at the same time, basically nailed the cause of lung cancer as being cigarette smoking. But in this, probably people mostly don't pay much attention to this, but right off the bat, he talks about how he came up with this study, and it was from descriptive epidemiology point of view that they had noticed there's a rapid increase in lung cancer over time, and they decided, based on descriptive epi observations, that there had two main causes that they brought this study to test. One was that the increase was due to general atmospheric pollution from the fumes of cars, from the surface of dirt and tarred roads, and the second was smoking. And as you all know, the smoking hypothesis won out. As Sir Richard told me and told anybody who wanted to know, he was not betting on that one. He was betting on the pollution one. And if you pressed him further, you had to press him pretty hard to go further. He had admitted that it may have something to do with the fact that at this time he was a cigarette smoker. So. This is a good example of time trends, and looking at things that change over time and looking for other things that changed in the same manner. The other standard one is place, geographic differences. I'm going to show you a much more recent slide, but this has been going on since the early 50s as well, looking at rates in different countries and comparing the highest to the lowest. And as you can see here, in this particular one, we go from a 5-fold difference for leukemia up to 155 for melanoma. This one is from relatively recently. It actually started back in the 1940s and 1950s with the few countries that are actually publishing data on this, and probably the most common was to look at East Asia versus the Western world. At that particular point in time, we had Bill Hensel was here, who could be considered the start of the descriptive epidemiology at NCI. He was a consummate statistician, but also a consummate epidemiologist. And he worked with Dr. Sagi and others, and Dr. Kudahara, looking a little more closely at the difference between East Asia and the U.S., and was one of the first to see that, I won't go into this in detail, that even with some small numbers and a lot of bouncing around, you could see that subsequent generations to the U.S. started to move towards the U.S. rate. He also, while he was at it, decided he might as well look at migrants from all over the place, and just went through and did a systematic look at cancer mortality from all sites by virtually, by where people came from in this country. It's kind of ahead of his time in this particular circumstance. I'm just going to point out this one where you'll notice that Czechoslovakians have a very high risk of GI cancers, particularly colon and rectum. The migrants from there to this country do. And I just put it up there because you're going to see it again in a little while. Whoop. So I don't know if you can see this very well or not, but after Bill Hunsel in the 50s, Joe Fraumeni showed up in the 1960s and immediately got interested in doing things a little more aggressively and following them up with analytic studies. This is from the picture from the mid-70s, looking at the cancer maps, which you'll see a heck of a lot of in the next few minutes. That particular one we're looking at, I'll talk about after Joe talks about a few others. The other individual here is Tom Mason, who was a statistician, who was a key person in our developing the cancer maps. And I also like this particular one because it shows that Bob Hoover has jet black hair while Joe Fraumeni doesn't. So I think with that said, I'll turn it over to Joe to talk about some of the interesting findings and led to interesting studies.
Yeah, Bob showed the bar graph for the international variation in cancer, which was very striking at the time. And we wondered what would happen in the United States, whether there was also geographic variation. And we looked first at the regions and state rates, and we didn't see very much. It wasn't very exciting. But then we decided to drill down into at the county level. And what we saw, what we thought was quite amazing. For lung cancer, for example, the rates were high in the Northeast, presumably due to the historical smoking habits. But the highest rates in all were in the Southeast Atlantic coast, from Virginia down through Georgia and into northern Florida. And we were struck by this, and we really didn't know the reason. So we sent Bill Bullock, volunteer, who's a southern boy, decided to go down, to drive down into this area, particularly down in Georgia, which had the highest rates of all, and take a look around and see if he could see something. It was in males rather than females, so we thought about an occupational exposure. And the leading suspect at the time, according to Bill, was the paper mill industry, and there was a fair amount of pollution from that. But we weren't sure about that. But fortunately, there was a county health officer who accompanied Bill on the trip, and he said, you know, there was something peculiar going on. Back during World War II, there was a very active shipbuilding industry and a tremendous amount of asbestos exposure. So we quickly launched into a case control study in Georgia, and this was followed by same studies in Virginia and also in northern Florida. And what we found in all three were an increase that we expected with smoking, but also a tremendous increase with employment in shipyards. During World War II, there was so much exposure to asbestos. But what was really striking was an interaction, a synergistic interaction between smoking and shipyard exposure. Now, if I can stop for a minute, when I showed those maps to Bob Miller, who was the chief of the epidemiology branch, Bill Hensel, as you saw before, was the chief of the biometry branch. And Bob said, yeah, that's very interesting, that dominant pattern along the eastern seaboard. But what about, look at the outliers. Look at those areas that had high rates out in the west. And he had this cover of the New Yorker on his wall. And he was fascinated by the New Yorker, and this may be why he was such a good writer. But he said, look for the outliers. So the outlier here, there were some in the western part of the country, but the one that was most striking was this one up in Montana, with very high rates, in an area that didn't have high rates, that had low rates. So what was going on? It just so happened that Anna Lee and I, back in 1969, had published a paper on a cohort study of cancer in Anaconda, Montana, and where there was arsenic exposure, which was the leading hypothesis for us. And there was a threefold increase in lung cancer overall in the plant. This chimney was the tallest chimney, smokestack, in the world at the time. To give you an idea of how tall it was taller than the 2 stories higher than the Washington Monument. And there was a lot of air pollution around there. So overall, a threefold increase in risk, a dose-response relationship with an 8-fold risk among employees who worked 15 years with heavy exposure to arsenic. And the results prompted new OSHA regulatory standards. We had to go down to the Department of Labor to testify. And there was a lot of pushback from industry-related epidemiologists who did not find in 2 previous studies an excess of lung cancer in smelter workers exposed to arsenic. But it prompted a reanalysis of the data, which showed results that were very similar to ours. Bill and I wondered what would be going on outside the plants. We saw a lot of air pollution there, and we knew, of course, there was an occupational cancer risk, but we looked at the surrounding female populations, using our cancer mapping data. We looked at the surrounding male and female populations in the counties around the smelters, not only the copper smelters, but also the lead and zinc smelters, which emitted arsenic. And we compared it to the counties that had other kinds of smelters that did not produce arsenic. And what we found among males and females, the females are shown here, was an excess in all population sizes of lung cancer in those counties shown in red. And then Bill wasn't satisfied with this correlational studies, and so he and Linda Brown went up to Pennsylvania, where there was a large zinc smelter, and carried out a case control study of of lung cancer, measuring the arsenic. I don't know, is this you, Bill? Measuring the arsenic in the dirt at the homes that were around the smelter. And there was a pretty strong relationship. So this pointed to the importance of neighborhood or community air pollution in addition to the occupational hazard. I should mention here that there was a very momentous event that took place in 1972 when President Nixon visited China. And this was a big deal because China had been closed and we did not know what was going on there, but it turned out to be an epidemiological paradise. And as an example, here is again lung cancer, female lung cancer in China. And what we saw from maps that were had just been published by Li Bing and her colleagues at the Cancer Institute in Beijing were very high rates among females in the Northeast, Manchuria particularly, where it gets pretty cold. And these are mainly adenocarcinomas, and they were virtually all non-smokers. So this was quite interesting to us. These are largely urban areas, but also Shanghai down here also had high rates, and Shenyang and Harbin up here in the north. So, but Bob Miller reminded us, what about outliers? And this was an outlier here, a tremendous outlier. Shunwei has the highest mortality rate for female lung cancer in China and probably in the entire world. It has been described as a Hiroshima event in China. And population laboratory was set up in this particular area. In Shunwei, there was tremendous household air pollution. And these are examples. You could barely breathe in the homes. This is our first study in Shanghai. We found an association with cooking oil fumes from high temperature frying. This is a wok with very high temperature cooking. And then in Shenyang, a couple of years later, we did a study in homes that had what was called Kangs, K-A-N-G. And Kangs were a fascinating apparatus that not only was used for cooking and for heating. And there were pipes that ran through the beds to warm the room and the beds. And these pipes leaked, so there was a lot of pollution in the household. And then also in Jiangsu, these were people who were called cave dwellers, and they lived in these underground caves that where there were high levels of radon. And the radiation epidemiology group with John Boyce and Ruth Kleinerman, Jay Lubin, carried out a case-control study and showed an association with radon levels that were as high as what was seen in some of the underground uranium miners, which had a high risk of lung cancer. But also there was a lot of household air pollution from, particularly from cooking, and cooking using rapeseed oil, the same kind of oil that was used in Shanghai. And this oil generated a blizzard of carcinogens including 1,3-butadiene. These were tested by Peter Shields and Kurt Harris at NCI, and also benzene and a variety of other carcinogens. So, Ching Lan, in our occupational and environmental epidemiology branch, has been on a hot streak since 1993 and up to the present time with a blizzard, or not a blizzard, but of studies having to do with environmental risks. And first it was case-controlled studies in Shunwei, which showed that smoky coal use was the primary risk factor. And this was compared with homes that use smokeless coal. Smoky coal is bituminous, and smokeless coal is anthracite. And the risk of people in homes that use smoky coal was 99 times the risk of those. This is smoky coal. They use chunks of coal, and the smokeless coal is a powdery mix. So the people in the use smoky coal had risks as high as 99 times the other coal. And then a retrospective cohort study was carried out, and those homes that vented the stones using chimneys or using portable stones that went in and out of the homes, the venting stone reduced the risk by 50-fold. And then there were a whole series of molecular epidemiology components that continue to this day. There was detailed exposure assessment that showed that there was probably a risk from the polycyclic aromatic hydrocarbons, as well as possibly the particulate matters generated by the smoke. And there was a unique gene expression signature, and there was a tumor tissue mutation pattern was also found. And then more recently there have been consortial studies in urban and rural populations. How many counties? 10 counties, 10 countries across Asia. And the findings there in terms of the genome-wide association studies and somatic mutations have been fascinating. And I won't go into that, just tell you that it's fascinating. And the studies in general have had an impact on the IRC monographs, which declared smoky coal to be a group 1 carcinogen, and impact on WHO indoor air guidelines, and on the global burden of disease study, in which household air pollution was from smoky coal was declared number 3, right after high blood pressure and smoking as risk factors. Okay, so I'd like to take a little trip down memory lane, if I could. In June 22, 1962 was the exact date in which I arrived at the Cancer Institute. I paid 20 cents for this Life magazine, not necessarily because of Marilyn Monroe, who I liked very much, but this feature article was said new evidence that cancer may be infectious. Now, at that time, cancer etiology was a black box. Really, nobody knew, if you really quizzed them, what was causing cancer. Smoking was under suspicion, and at that time, the Surgeon General was convening an outside committee to review the evidence, which didn't come out until 1964. That smoking was a risk factor, but it was a black box. But the Cancer Institute was convinced that infection was the main cause. And infection, was accounting for most, if not all, of cancer. And the Institute was mobilized to investigate this hypothesis. In the article, what I found particularly interesting, I didn't know a thing about cancer epidemiology at the time. I was really naive epidemiologically, and there was a house that was pictured. It was sort of a ghostly photograph that referred to this house, which was a cancer house, in that all the occupants, nearly all the occupants, who were either related or unrelated, had cancer. And outside the house was what was called a cancer tree, which were excrescences on the trunk of the tree caused by an infection. And nobody knew quite what the infection was, but it was thought this was related to the cancer house in some way. And there was another house down the street here that had the same configuration of cancer house, cancer tree. But the article really summarized all the clues that were coming, that particularly from the laboratory, that cancer was due to an infectious process. Now, right after that article came out, there were reports that SV40, simian virus 40, was detected in cultures of rhesus monkey kidneys that were used to produce polio vaccine. Now, this raised a lot of eyebrows, and then in 1962 at NIH, Bernice Eddy and her colleagues reported that SV40 is a potent carcinogen in laboratory animals. It started with newborn hamsters, and then it spread to other kinds of laboratory animals. So this was a startling discovery and raised tremendous medical and public health concern. It was estimated that perhaps 10 to 30 million Americans may have received SV40 contaminated vaccine from 1955 to 62. And there were reports that subclinical SV40 infections occurred in humans and that there were transformations resembling neoplasia in human tissue culture. So what to do? There was a big conference at NIH that was led by Mike Shimkin, who was head of the what was called Field Studies and Statistics, which included the biometry branch and the epidemiology branch, which was very small, with just Bob Miller and myself at the time. And Mike Shimkin, the evidence was revealed that there was SV40 contamination of polio vaccine that was used in the mass vaccine campaign in 1955, and that these lots of vaccine, the batches, varied by state. In the north, there were high levels of SV40, and then there was an area, multiple states that had low levels, and then there were a number of states, particularly in the south, that had no SV40. So it was thought maybe a comparison could be made of the cancer experience between these groups. Now, the dose was given to children who were 6 to 8 years old in the first and second grades in a short period of time in 1955. Now, Mike Shimkin, I was sitting in the front row around the table, and Mike Shimkin turned and said, "Joe Fraumeni is going to do this study." I didn't know what the devil was happening. And fortunately, I was sitting next to Fred Ederer, who was in the biometry branch and a seasoned statistician, and he said, "I'll help you." So we conducted this comparison in the form of a developing statewide cohorts of children who received the polio vaccine according to the levels of SV40 contamination. And what we found, it was a short period of time, but there was no difference in the cancer mortality experience in the states that had high levels versus low levels versus no SV40, both in terms of leukemia and all other cancers except leukemia, and these were all different sites of cancer. So that's it for now. Thank you.
He's coming back, don't worry.
Not for long.
Okay, so this is the map that I was holding in that picture you saw some time ago, and this is actually the county map or state economic area, which it is for cancer of the oral cavity and pharynx. When we first looked at this, it was just stunning that this huge concentration in the southeast, and this is in women, and it was essentially confined to women, not in men, which was doubly confounding. Usually when we saw gender differences, it was in the males being higher, and we would pursue it for occupational reasons. So this was quite provocative, and about almost exactly the same time, there was a publication from England of oral cancer in female textile workers. And so, of course, Bob Hoover had this bright insight in that textile work used to go on in New England, but some time ago in the early to mid 1900s, it moved south for financial reasons, so that there was a fair amount of textile work in the South, and those were mainly staffed, mainly female workers. So figured that this, well, we had a good hypothesis. But Bill Blott was interested, and there was a young woman who was finishing her PhD at UNC who was looking for a thesis project, and her name was Debbie Wynn. I don't know if this Debbie is here today or not, but there she is. All right. They came back and said, well, we should start this study, but there's something else that goes on in North Carolina and the Southeast that doesn't show up in any of the books we were looking at for hypotheses. And I said that the women there use actually a lot of snuff. They dip snuff. And I said, well, that certainly was interesting. You have oral cancer and you're dipping, putting tobacco in your mouth. But yes, actually proper older ladies in the South, East at the time, would never touch a cigarette, but they would dip snuff. So the analytic study there certainly did indict snuff dipping, up to about a 50-fold excess risk for long-term use of snuff. This was a particularly heartening study in terms of its immediate impact. At the time, advertising cigarettes in the media and television and radio had been outlawed, but not snuff, because it wasn't considered to be a carcinogen. This finding that Bill and Debbie had was so impressive that it led immediately to changing and to including the exclusion of advertising on media for this too. This is Walt Garrison, who was a running back for the Dallas Cowboys, encouraging you to put some of that in your mouth. And this was a young man who followed his encouragement and was using it. It's also another sort of notable difference between Joe and I. Joe shows you a picture of Marilyn Monroe in her robe. I show you a picture of an ugly football player putting tobacco in his mouth. Joe showed you a couple of examples of the unusual observation, so I'm going to follow up with another one. This is colon cancer, and I should have got, this is at the county level. I should have put the state economic area, which groups them a little bit better. But in point of fact, it was mainly a northeastern disease in terms of where the excess was. So it was immediately catching your eye was a couple of counties in southeastern Nebraska that showed up in both men and women. And this is mainly just farms and small towns. It was really so striking and occurring in both genders. We thought it was worth looking into. Linda Pickle, who was with us then, was took on the task, and she looked through all the information from the census and everything else that we could get, and she said, you know, it just looks like a regular old farming area, no different than the farming areas in the Midwest, except for one thing. She said that, odd, they have a really high number of migrants from Czechoslovakia. So the man to my right, who never forgets anything, that obscure table and Bill Hensel put out that had almost dozens of, he said, didn't Bill Hensel find an association? And I said, by golly, you were right. So we went to do a field study there, which was very difficult because there's not too many folks out there. So the sample size was not generous. But in fact, it nailed it. The reason was that all of the excess was in the Czechoslovakian migrants, and actually it looked like Moravia was probably worse than Bohemia. Unfortunately, because of the sample size, we couldn't figure out what the exposure was. They basically, anything that you'd associate with being Czechoslovakian was associated with excess risk because it was, you were Czechoslovakian. So we did not, we were not able, and just to show you that in more recent times, Czechoslovakia still leads, at the time Bill did it, Bill Hensel did it, we didn't even have data for Czechoslovakia, but they lead the world in this one's for rectal cancer. So if anybody has a great idea about why Czechs, and maybe specifically Moravians, would have, I can tell you one thing, that while we were there, Linda was able to determine that it went away really rapidly with subsequent generations. So it does look like it's probably an environmental exposure, but a little bit unclear at this point. So anybody have a good idea? The next one is just kind of a cautionary tale for descriptive epi. Actually in the 50s, it was starting in the 40s and in the 1950s and early 60s, about the biggest, the most enthusiasm for looking at differences in cancer were international differences, and the most prominent one was looking at breast cancer in Asia versus the U.S. I showed you before the thing that Bill Hensel had looked at migrants from there. This one is quite a bit later, but in fact this hypothesis and a very similar curve was first produced in the mid-1950s and early 1960s to show this tight relationship between dietary fat and breast cancer risk, which everyone accepted as probably the reason for it. And well, and it was also noted that the U.S. women had higher estrogen levels than Asian women, so it was determined. It was the international differences was due to dietary fat operating through estrogen. And nobody did anything more because we had the answer. Over the next 20 so years, 30 years, more and more evidence came that dietary fat didn't seem to be related to cancer risk in any population. And if you actually sat down and did the math, which I don't think anybody did until about 10 years ago, that the difference in estrogen levels between the two populations was impossible to cause the differences, the magnitude of the differences. So the cautionary tale is sometimes descriptive epi can be so seducing that you believe it without ever testing it. This one provoked us, so we took advantage of the fact that more registries had come online, so you can actually see this gradient here between the US being the highest, but we have a new low here. Used to be Japan, then it went to China. Mongolia actually has a rate which is about 1/3 that of China, which was provocative. These are fairly recent data, but they have a totally different lifestyle than the Chinese. Their main source of calories is meat and dairy, and you can determine here that they exceed the UK in those categories. So the dietary hypothesis didn't seem to work too well in this particular circumstance. Looking at hormones, I'm probably not going to be able to read this from here, but I believe this one is, help me out here, is this estrogen E1 here. Actually, the Mongolians have higher estrogen levels than British women. British women have higher androgen levels, testosterone levels, and the Mongolians also have higher progesterone, particularly in the follicular phase. So I think if you were actually generating hypotheses now, you'd say maybe hormones do play a role, but it ain't estrogen. Looks more like you're more interested in androgens and progesterone. Again, just a kind of cautionary tale that if you get a hold of something and it looks interesting, it probably is well to pursue it pretty aggressively. These were obviously geographic comparisons. Joe's going to show you one, and I'm going to show you one based on descriptive studies based on time trends. And I'll give it to you.
Yeah, Bob showed the trends for lung cancer, which were so intriguing and raised the hypothesis that smoking may account for the epidemic of lung cancer. And another epidemic that has occurred, thanks to the analysis from the SEER program, which made it possible to distinguish between two major types of esophageal cancer, squamous cell and adenocarcinoma. It was thought before the SEER program came out that that esophageal cancer was mainly the squamous cell type, and what it shows here is that it was, but then there was a crossover then in the 1980s, and adenocarcinoma shot past squamous cell carcinoma in white males. Also, this shows another exciting aspect of descriptive epidemiology, and that is the racial disparities that can be gleaned. Before the SEER program, you really couldn't distinguish blacks from whites. There was non-whites and whites in the periodic cancer surveys, which had previously been carried out in 3 time periods. But it was amazing to see the pronounced difference by race, and then the sharply rising incidence of this particular type of cancer. Now, what accounts for this? The major risk factor for adenocarcinoma, I think, there's general agreement that gastrointestinal reflux disease, what's called GERD, is the primary risk factor. But there are a lot of other risk factors that contribute to this, and one of them is obesity. And there was in males a study by Linda Brown and Bob Hoover in 1995 showed a dose-response gradient rising to 3-fold among the chubbiest members of the population. This is in white men. Subsequently, we carried out, this is a study led by Wang Ho Chow in 3 areas of the SEER program, Seattle, Connecticut, and New Jersey. And this showed the gradient occurring in both males and females. And there were other risk factors which I won't go into, but it shows just shows the ability of descriptive epidemiology to generate hypotheses. Bob?
Okay, I know it's closing down towards 3:30 here, and we actually can stay in this room for another half an hour, but if I'm going to try and just zip through the next example to get to the message at the end here. But this is a more contemporary example. This is work that's done by Bill Anderson and Phil Rosenberg relatively recently, and basically just shows that in the past people have just looked at breast cancer. If you take a time period when you can actually split now what the 2 versions of it are, ER positive and ER negative, you'll notice that they're doing different things. ER positive is going up, ER negative is going down. And this brought to mind the fact that there was a real anomaly going on for decades, from the late 30s through the 90s. Breast cancer incidence rates were going up at a fairly steady rate. Breast cancer mortality rates were flat. We never figured that one out. It's possible, right? You can't go back and do typing on historic, but it's possible that the more aggressive ER negatives were going down over that same period of time. Well, they may have started much higher and were canceling out in the mortality column any excesses that would be there from the ER positive. Just a quick note: if you get it down to this level too, you can see some other interesting things. This is ER positive. ER negative is just going down. ER positive is bouncing around a little bit, but if you'll notice, right, this decline right here happens about 1 to 2 years after the WHI trial indicting estrogens and replacement therapy, and the consequent dive in the incidence. And it had been noted previously that menopausal hormone use, you could use it for 15 years and get up a pretty high risk. If you stopped about 2 years later, you had a risk as if the women had never used it. So it was a nice little example. You know, we had done the analytic studies before, so we knew what was going on, but it was a nice example of what you could be provoked by. So what I wanted to get to is that descriptive epidemiology, I think, have been remarkably productive for generating insights. And we haven't exhausted the well. In fact, the well is overflowing with opportunities. There have been massive changes in geographic patterns and in time trends that nobody has evaluated. You can drop by and you can use SEERStat. You can do a lot of ways of looking at this. And I mentioned, now that we have all new disease classifications based on biologic characterization, it's maybe a little early to do too much extensive, but I think it gives us the opportunity to look at breast cancer subtypes, even esophageal adenocarcinoma, which splitting from squamous gave us a lot of information. Now there are 3 mutational subgroups, which would be pretty interesting to look at. And I should say that what we've been talking about is descriptive epidemiology, which is a discipline which is not solely, but largely done by governments. By epidemiologists working for government and international agencies. That's mainly because it's not hypothesis testing. And if you write a grant to say, I'm going to look for something rather than have a hypothesis, you don't get funded. So most of what I've showed you is the result of national entities, both in Britain and in here and elsewhere, mining the descriptive epidemiology opportunities, something that we think we should continue to do. And my own enthusiasm is there's been an explosion of new data resources. Every time I turn around, somebody's telling me about some data that somebody has put on it, has measured and put out there and I don't even know how to figure out how much is out there, quite frankly. To be a good young person who follows who follows these things. It's also possible to get a more refined level of data. You can get them at the block level rather than the county level. A lot of these, and there's new methods developed for analysis of so-called big data. This one is a list of relatively new general kind of environmental databases that Rena Jones was nice enough to provide for us. And I think it's even longer than this, because again, we're hearing that gentleman from NOAA that came to talk to us and talks about what NOAA has collected with respect to air pollution and the level at which you could probably identify your house if you wanted to know what your air pollution level was. So there's these are some of the relevant lifestyle databases, and again, there are way more than this. I'm just not even aware of. A good place if you wanted to start at a place. Just going to SEERStat, they've done a nice job of taking a lot of census data and making it really easy to marry up to incidence data and to analyze. So I also have expanded my definition of descriptive epidemiology to include linked health databases. Technically, it's not it's individual data. You have individual data about who had this particular disease and who had subsequent cancer. So it's technically not descriptive epidemiology, but in reality, these databases don't have anything else to let you control for. So practically, they're like descriptive epidemiology, and they are proliferating. You know, Eric is in the front row here, and he's made a lot of very important and interesting observations with the HIV cancer registry match data. Transplant registry, I dabbled in back in the 1970s, has now gotten to high energy level of what you can actually look at, the UK clinical database. So again, the combination of what's become available, the value of descriptive epidemiology, and the opportunities, and the fact that I think we're supposed to be doing it. We're a national organization with opportunity to tap into these other national databases. So I'm trying to gin up a little enthusiasm. Back in 70s, 80s, and in the early 90s, most of our fellows were doing something in the way of descriptive epi. They may have been focused in a particular area, but if their interest was in a particular cancer, we sent them down the hall to do descriptive epi of that cancer, along with all of the other things that were going on. So this is the end of my pitch here, but I'm pitching for this. I think there are a lot of great new opportunities that I couldn't even have imagined back in the early 1970s, that to help us gain testable hypotheses that we can then take into the field, and so that's my apostolization for today. And Joe and I want to thank you very much, and you'll be here for as long as you want to ask questions or discuss things. Any questions?
I'd like to thank Meredith Shields for inviting Bob and myself to give this seminar, and also for taking the lead in organizing the next generation of studies in descriptive epidemiology. And also thank Mary Ward, if she's here, and Rena Jones for organizing working groups in descriptive epidemiology, particularly in geospatial opportunities that have arisen. And thanks to Stephen for his support in this particular area.
Be happy to talk about anything you want. Silenced the crowd. All right.
Thank you very much. Really was wonderful. Joe, I wanted to ask you specifically about the cancer maps. Okay, you know, this was something that you and Bill and Bob had done years ago, and they've evolved. The question is, what do you see as really the next important step in revisiting them? As Bob has pointed out, the importance of looking at things. What kinds of questions do you see as really the next burning set of questions that could and should be developed? And not only the scientific questions, but are there infrastructure issues that we need to address or put in place to be able to most effectively ask these kinds of questions?
Well, I think first of all we have to look at the updated cancer maps. I'm not sure we have as yet. We know that they're just in the atlases that we created, that there were tremendous shifts in the patterns, spatial patterns of cancer, and if that lung cancer map, for example, which showed high rates in along the East Coast, a lot of that has faded. In fact, it's moved inland, and the rates are highest in across the Southeast in Appalachia, and so we really have to look at it. The other thing that has really been fascinating is the racial disparities. We looked at our first study was on American Indians, and then we just looked at every single racial group that we could glean from the death records that were kept at at what's the name of that agency? Huh? Yeah, the National Center for Health Statistics. And that was not an easy job. And then, of course, the studies of Alaskan natives were particularly interesting. And so, and moving from mortality to incidence and comparing the two has also, I think, is the next, one of the next steps. And I think a lot of this is now being done. And thanks to our colleagues in the DCCPS and the fact that we are collaborating with them, I think has also raised the opportunities.
Thank you so much for your presentation. I'm wondering about, you had asked about the well is definitely not dry. I'm wondering also about how it's changing. So especially in the United States, we know that racial groups are no longer distinct. We have a huge proportion of Americans who are replying on the census that they are more than one race. And how do we account for this, and how do we measure going forward, and who decides what bucket they get placed in order to tease apart differences that may happen by race? I'm wondering if you have thoughts on that.
Well, as usual in epidemiology, we're beholden to the people who make those rules. So it's the census people who decide how they're going to ask people to designate themselves, and they are the ones that now I've seen just in the past few weeks, I've seen the opportunity to look at geographic differences and take in and out white Hispanics, because white Hispanics are whites if you look at it, and it makes, actually for some cancers, it makes a major difference just by if you move them in or move them out of taking a look at whites. So I think it is a tool, and as long as they give us some opportunity to distinguish, then we can use it. If we have to go out and generate our own means, we will not get it on the scale that we can get it with the.
So I have a question for Joe. The cancer maps are a powerful, and have been a powerful tool for identifying predominant patterns, whether it's the Mississippi Delta or the Georgia coastline or something like this. My question is about the Bob Miller approach of looking for the outlier. Among many possible outliers, how do you choose? How do you choose the possibility of not going after some, wild goose chase and coming up with something? Is it just general knowledge of what's out there?
Well, I think you've got to have some ideas. You have to know what the you have to know the literature, and I think first of all, and Bob, do you have anything to add to that? Yeah.
Well, I can assure you that there are tens of thousands of isolated observations that we haven't pursued. Just by looking at those maps, you can tell. So yeah, I think what we look for, it was with the one in Montana, it was so odd sticking out there like it was, and him having just done a study of the occupational, that was like a no-brainer. We would go there. We already had a hypothesis. They had this huge, and maybe it was a general environmental as well as an occupational. So that was fairly straightforward. The Nebraska colon cancer was, again, it was in both men and women, so that actually was somewhat of a hint that maybe it wasn't just random. And then we had something from about 30 years prior telling us that maybe Czechoslovakians were at. So I think I think part of it relates to identifying what might be an outlier, but then a part of it is investigating the credibility of the rate itself, as well as is there anything, a hint of anything that might be worth looking at further. So I think that's kind of the way we've done it in the past. But.
Yeah, I mentioned the fact that we knew so little about cancer etiology at the start of, at least the start of our branch, but and the fact that infections were the major and the most popular hypothesis at NCI. And there were some areas, genetics as an example, and nutrition as another example, they were really specialties that were boutique, and very few people at that time were working in those areas. So, but as we have progressed, and we now know a lot about what causes cancer, and we have to use that knowledge to decide what to go after, what is just occurring by chance alone, or what is ripe for study. And I think the opportunities are just enormous at the present time. I just would encourage people to mine the data that Bob Hoover presented. It's just been barely touched upon, I think, so far, and I think the opportunities are expanding. The resources are unmatched, particularly here at NCI, and we should take advantage of them.
Fabulous talk. As someone who was kind of a bystander to a lot of this stuff as it was going on back in the day, I still get goosebumps when I see the kind of observations and findings that have influenced public health, that have emerged from this kind of work. And so I fully support your urging us all to reconsider this as a research domain. The problem is that it is, how do we support this kind of work within the division, given the kinds of research priorities that you yourself have identified that interfere with this sort of work being funded in the extramural community. This proposals that come forward within DCEG trying to follow these leads are up against the hottest new molecular hypotheses, and we've got a new next generation cohort study coming that's going to absorb a significant amount of resources and energy. How can we structure the division in a way that makes sure that there are resources available for the right kind of questions to be supported?
Yeah, I can't tell you how to restructure the division, but I can tell you that at least in my experience from 1972 onward, it wasn't easy getting money to do the field studies we did do. We had to compete against a whole lot of other good ideas that people wanted to do in the division, and it relied a little bit on the impressiveness of our hypothesis and what led us to that. And in the ones I've showed you, it was pretty persuasive that something was going on that could be detected by indirect methods, and that we had some very good hypotheses about what might be explaining it. So we tended to do pretty well, but not universally. There were some field studies we did not do. We published the descriptive data and didn't, and hoped somebody else would follow it up. But so I think it is just as it always has been. You got all sorts of good ideas come from all sorts of people, and you have to try and make the best judgment you can about which ones to fund and which ones not. I think it's the advantage you have here is that you can generate the hypothesis using existing data and your imagination, and it actually is a pretty cheap way of coming up with some good ideas. Then you have to kind of sort them in your own mind about which ones are worth pursuing aggressively and not. But
I think also the trick is not to think of molecular genetic science as being distinct from epidemiology. It's now increasingly important to merge the molecular science and genetics, population genetics, with epidemiology, and I think that's the future, and not to consider that we're necessarily in competition with the laboratory scientists, but that we're increasingly collaborating with them.
Why don't we have one final round of applause and thanking our speakers for more than just speaking today.